In addition, I again forgot to hit record on Monday for our class. So I will have to re-record that one as well. So the ones that need to be re-recorded will happen. They just have not happened as yet. If anyone ever happens to do audio, their own audio during the lecture, let me know, because that would actually be quite helpful. Last time we talked about NK cells, and we started kind of dealing with some details of NK cells. And if you recall, as we were talking about NK cells, we first had this idea of thinking about what was key for innate versus adaptive immune responses. And we thought about timing being different. So we've got early timing being important for innate responses, where we had some later timing for adaptive responses. We had the requirement for VDJ for adaptive, where we had germline receptors, meaning they were just regular old genes encoded in the genome. For innate. For adaptive, we had memory, as well as the general clonal expansion. Parts of the response. With innate, we see kind of the same response every time. And with the innate, we saw that we usually had cells of the myeloid lineage, whereas with adaptive, we usually have cells of the lymphoid lineage. And then when we looked at NK cells, we noted that those NK cells are cells coming from the lymphoid lineage. They are cells that respond pretty early. They're cytotoxic. In the same way that CTLs are cytotoxic. And they have their responses enhanced by type 1 interferon. Enhances activation, or enhances response. How about that? Enhances response. So we started to see some features of NK cells last time that we could note, kind of made them straddle the line a bit of innate versus adaptive that we had been talking about before that. And we left off talking about NK cell receptors. So, as I told you last time, NK cell receptors can be divided into some different groups. One grouping that we can use for NK cell receptors is that they can be in different structural families. So there are some that are called KIRs. There are others that are of the NKG2 family, and so those are shown here. You can see they're sort of shown as either proteins with these rectangles, which are showing us immunoglobulin-like domains, or these little bean things. We can also divide the NK receptors into classes of activating receptors or inhibitory receptors. As you might guess, if you engage an activating receptor, you turn on the NK cell. And if you engage an inhibitory receptor, you turn off the NK cell. You should notice that there are members of the different structural families in each class. So it's not like you can say, well, KIRs are all inhibitory and NKG2s are all activating. There's sort of a mix of each kind of structure in each of these two classes. If you notice, the activating receptors all pair with a partner protein, similar to what you saw for T cells with CD3 and what you saw for B cells with Ig alpha and Ig beta, also known as CD79A and CD79B. Officially this protein is called DEP12, so that's actually, don't worry, DEP12 being the name. The key thing to note about this partner protein, officially DEP12, is that this partner protein contains ITAMs. And you've seen ITAMs previously in the course. This is a string of amino acids containing a tyrosine. And this particular string of amino acids containing a tyrosine, if you phosphorylate it, leads to an activation signal. So ITAM, if you remember, stands for immunotyrosine-based activation motif. Alternatively, the inhibitory receptors have a different domain in their cytoplasmic tail. That domain does not contain an ITAM. Alternatively, that domain contains something called an ITIM. An ITIM is another stretch of amino acids. It still contains a tyrosine. But if you phosphorylate the tyrosine in an ITIM, you get an inhibitory signal, an off signal, instead of an activating signal. And so while ITAM stood for immune-based tyrosine activation motif, ITIM stands for immune tyrosine-based inhibitory motif. So we've got these two classes of receptors on NK cells. You can also see the same information in this table from your textbook. So there are different classes listed here as different families of NK receptors. In many cases, the families have both activating and inhibitory members. And some examples are shown here. You can also see that there are some differences between mouse and human here. The NK cell receptors are encoded by genes that are on a couple of different chromosomes in human. Some are on chromosome 12, some are on chromosome 19. These are not subject to BDJ recombination. You just have a section of chromosome 12 and a section of chromosome 19 that have a whole bunch of NK receptors. There's no VDJ. These are all germline encoded receptors. And so again, if we think about the NK receptors, these receptors are germline encoded. So there's another place where we can see that NK cells are maybe not behaving as nicely as we might like. We were able to start to understand the details of NK cell receptor signaling and how and exactly what the deal is with NK cell receptors in a pretty famous experiment, which is shown here, done in 1996 by someone named Klaus Carr. I'm going to draw out this experiment on the board. There are a couple of places where you're going to look at what I write on the board and you're going to say, she's being a little over the top in describing things. Like there are some things where I'm going to write more words than you might expect. The reason is that there are a couple of places where one can get very confused with this experiment. And so I'm trying to avoid confusion. So make sure you're kind of paying close attention to some things I've stressed, because I'm trying to keep you away from confusion. I will also tell you, I think this might be the first time in a lot of years that I've done this one without notes. So, like, this is a tricky one, but we can handle it. Okay? No, we don't need a drum roll. Okay, so they started out this experiment with some mice. These mice were of the H2B haplotype. I don't actually remember which one they were, but it works if they're H2B. And these experimenters did an experiment where they did a transplant into these mice. And they transplanted some H2B tumor cells. Into this mouse. When they did that experiment, the tumor lived. The mouse got a big giant tumor. And the mouse died of the tumor. The key thing here, the first time I ever tried to take notes when someone described this experiment to me, I just wrote lives and dies. And sometimes the thing that lives is the tumor, and sometimes the thing that lives is the mouse. And so I was so confused. So make sure when you're thinking about this, you're paying attention to, is it the mouse or the tumor that's living and dying? Okay? So here, the tumor lived, the mouse died. They then also did some different experiments where they used a mouse that was of the H2D MHC type and put in the same H2B tumor cells. When they did this experiment, the tumor is killed. The mouse lives. Happily ever mouse after. And we now know, and this will be subject of some stuff that we talk about later in the semester, that this is actually due to a CD8 T cell response. This is similar to transplant rejection. This was part of that experiment that they did. And part of what they saw, or part of what they were thinking when they did this experiment was, oh, this is really cool. You know, we can think about immune responses to cancer. And again, later in the semester, we're going to talk about immune responses to cancer. So they thought that was cool. They wanted to sort of try to understand that. And one of the things that they did is they made some mutations in their tumor cells. So they took their tumor cells and made some changes to them. And they made a specific mutation on these tumor cells that we now know. They didn't totally understand exactly what was going on, but what we now know is that they took the same tumor cells, but they mutated them so that the MHC class 1 particularly, but the H2B molecules were not on the surface of the tumor cells. The official mutation we now know is they made a mutation in TAP so that no MHC class 1 could get to the surface of these cells. That's actually, they didn't know that then. They just knew that the cells were H2 deficient. So they knew that the class 1, all the MHCs didn't go to the surface of the tumor cell. They didn't know why. We now know it's because they have a mutation in TAP. Yep.
Was that into the H2B or
We're getting there, yes. So first they took these tumor cells that do not have the H2B molecule, but otherwise they are the identical tumor cell that they used before. And they did the identical experiment, which is that they put them into the H2B mouse. And so you can see the experiment is basically identical, except for one thing about the tumor cell. And when they did that experiment, they found that the mouse lived and the tumor died. So somehow, this tumor got killed with only one change, which was that it was missing MHC. Suddenly, the mouse's immune system could kill that tumor that it didn't used to be able to kill. We've only made one change in this tumor. It's just that it's missing MHC. They could do experiments where they basically tried to put the MHC back in and all sorts of fancy things, and they all worked as expected, with the idea that basically, in some ways here, it was almost like the MHC was keeping the tumor from getting killed. Once the MHC was missing, now the tumor could get killed. And so they came up with this hypothesis known as the missing self hypothesis. And the idea with the missing self hypothesis is that you have some part of your immune system, I didn't know what it was, that recognizes when a self cell, because these are basically self cells, are missing MHC. So if MHC is missing, you can recognize that and kill those cells. And so now what you've done is you've made these tumor cells be missing MHC, and suddenly they are subject to this killing mechanism that they were not subject to before. So that's what they got from this experiment. It led them to the missing self hypothesis, which is correct. And what probably will not surprise you is that the mechanism that they didn't really know about at that time, but we know now, was NK cells, because I'm telling you about this during the NK cell lecture. I know, sometimes I come up with these things. So if we think about it, we can imagine why it might be important to have some way to recognize cells that are missing MHC on their surface. Why could it be important to detect cells that are missing MHC on their surface? In particular, well, all right, we'll deal with that part in a second. Why would it be really important to have a way to detect cells that are missing MHC on their surface? Let's see, Jamie.
If the cells aren't functioning properly, you don't want to keep them around.
Okay, you don't want to keep a cell that's not functioning properly. Sydney.
There's a variety of viruses
it's super, super common for viruses to try to mess with MHC class 1. And so very frequently, when a cell is infected with a virus, that cell might lose MHC class 1. This is particularly true of some of the large DNA viruses like herpes viruses. The cell is going to lose MHC class 1. And the idea is that the virus is trying to kind of hide from the immune system. And this is such a common strategy in our back and forth evolution against viruses that we have a mechanism that actually checks cells, and if they're missing MHC, we sort of have this mechanism to say, you must be a problem. There are other reasons why cells lose MHC. Largely, they are because the cell is under stress, because it is either trying to replicate really fast, and reproduce really fast, or under some other kind of stress, or it's because it's making a whole lot of viral proteins. Sometimes it will lose MHC. But the general idea is if a cell has lost MHC, it's either infected with a virus that is actively trying to get rid of that MHC, or there's something else really wrong with that cell. And so the way that this works is that we have a whole bunch of receptors that bind. Oh, wait, sorry, before I get to this, the other point I was going to make. It actually works on this slide too. We have a whole bunch of receptors that are inhibitory NK receptors that bind MHC class 1. So the idea is, if a cell has MHC class 1, that MHC class 1 is on the surface, that turns any NK cell that comes by off. It says, I'm okay, you don't need to kill me. And so the NK cell is turned off. We inhibit that NK cell because we have proper MHC class 1. So the majority of the NK inhibitory receptors bind MHC class 1. And the idea is this is how we actually sort of detect missing self, is that if we have proper self-MHC, we inhibit NK cells, we turn them off. Notice that if we look at this list and think about the ligands for the NK inhibitory receptors, you see MHC class 1 a whole bunch on the list, but you don't see MHC class 2. Why do you think it is that we don't kill cells that are missing MHC class 2? In fact, that's true. We only kill cells that are missing class 1. We do not kill cells that are missing class 2. So this whole missing self business is really only about class 1. It's not about class 2. Yep. Yeah, only professional antigen presenting cells have class 2 on their surface. Most cells don't have class 2 on their surface. So if we killed everything that didn't have class 2, well, then we would just be a sack of B cells, macrophages, and dendritic cells, and thymic epithelial cells. That would be kind of not helpful. So this whole idea of missing self is really only about class 1. Because remember, all of our nucleated cells should have class 1 on their surface. And so if a cell is missing class 1, it will be killed by NK cells. If a cell has class 1, we inhibit the NK cells by engaging an NK inhibitory receptor. And so generally what we will see is we see something that looks like this. There are lots of different NK inhibitory receptors. The first ones that were really described were in the KIR family. And if we look at KIRs and how they bind to MHC class 1, we see something that looks like what is shown on the right here. So you can see the MHC in yellow and the peptide in red. If you remember, I showed you a similar picture to this when we talked about T cell recognition. And I showed you kind of what the T cell sees. There was a dotted box at that point, and that dotted box looks like this dotted box that's right here. So you can see the sort of angle that the T cell is binding at. And so what you'll notice is that the T cell is making some contacts with the yellow and making some contacts with the red. The T cell is seeing the peptide and the MHC together. The KIR is binding with this other solid box. And what you should notice is that is largely contacts with the MHC molecule. It's not really so much contacts with the peptide. So the KIRs don't care about the right antigen or the wrong antigen. They're just binding, is MHC there or not? They're just making contacts with the MHC molecule in order to see if MHC is there or not. There is one other there's some other NK inhibitory receptors that are a little different than this. You can see that this tells us about one example, but I'm going to tell you about a different example, NK inhibitory receptor, which is called NKG2A. And so this is an example that is a little bit different than the standard bind MHC class 1 kind. So this is one other kind of MHC or NK inhibitory receptor. When I was in grad school, I had the hardest time remembering this, or remembering part of this. And I had a friend who worked on NKG2A as well as another one called NKG2C. And I could never keep them straight. I could never remember which one was which. And she told me a hint. And her hint was, A should be activating, but annoyingly it's not. So it's the other way around. You want A to be activating, but that's wrong. So in fact, A is inhibitory. And that's how I learned to remember it. I know that's weird, but that's actually how I remembered it, is that, Carrion's receptors could not be easy. And so I'm going to be telling you about NKG2A. NKG2A is not in this immunoglobulin-like family. It has a somewhat different structural family. And in thinking about NKG2A and how NKG2A works, we need to remember a piece of information about cell biology. When we have a protein that is a transmembrane protein, that protein gets biosynthesized into the ER. Remember that? We've seen that before, that we have to have proteins biosynthesized as transmembrane proteins in the ER and then going through the secretory pathway. The way that that works is that the very first set of amino acids, I'm slightly oversimplifying some cell biology, but it's fine for our purposes. The very first set of amino acids are like a tag that says to the ribosome, hey ribosome, go translate this at the ER, please. So the very first amino acids actually make this move to the ER and get translated to the ER. And that's all those amino acids are used for. You might have heard of them before as a signal sequence. Here they're called the leader sequence. It's the same thing. It's just a set of amino acids that makes sure that the protein goes to the ER. And once the protein's in the ER, that signal sequence gets cut once the protein is finished. So we've got this, all of our MHC class 2 molecules, like HLA-A, get biosynthesized in the ER with a signal sequence, and that signal sequence gets cut. It turns out that we have a protein called HLA-E. If you look at the name HLA-E, what does HLA-E sound like to you? What kind of protein, if you just sort of heard that name out of context, what would you guess that HLA-E was? Yeah, Jamie? What does HLA-E sound like? Yeah. It sounds like an MHC class 1. Has HLA in its name and one letter. Sounds like MHC class 1. It's kind of like MHC class 1, but it's super special. It only knows how to present one kind of peptide. And the one kind of peptide it knows how to present is the signal sequences of all the other MHCs. So you can see HLA-E right here presenting the signal sequence, the red dot, from all the other MHCs. And so we can present that signal sequence on HLA-E. So it doesn't really count as a real MHC class 1 because it's not presenting pathogen peptides. It is only presenting signal sequences from other MHC molecules to turn off NK cells. And the idea here is that because we're presenting the signal sequences of MHC class 1 molecules, we're basically showing the NK cell, hey, I'm doing MHC biosynthesis correctly. And so it's sort of a check for biosynthesis of MHC molecules. And so kind of the same concept that we're basically saying, hey, yes, this whole MHC class 1 process is going well. Now we're showing sort of biosynthesis intermediates instead of actually showing the full-on presentation. But it's the same idea of we're turning off the NK cell because we're doing MHC correctly. So this seems great. And this is sort of how you can imagine what we've talked about so far. And so you can see that if we have a normal cell that has normal MHC, that MHC is going to bind to an inhibitory receptor to turn off the NK cell, and the NK cell is not going to kill. And if we have a cell that lost its MHC for some reason, we now don't have MHC in it, the NK cell has nothing to inhibit it, and so the NK cell is going to kill. So this is kind of what we've seen with the NK cell so far. Unfortunately, it's a little bit more complicated than this, because this assumes that NK cells are on and ready to go all the time. That's not really true. Sometimes when I show this slide, students think of a terrible problem, and they get very worried. Based on what you're seeing here, especially based on what you're seeing right here, can you imagine any problems? Are there any things that you're now very worried might happen? Sebastian? Yeah, what about red blood cells? Remember, red blood cells don't have class 1. If this was the whole story, we'd kill all our red blood cells. Now, if you remember from the first lab when we looked at blood under the microscope, there's a lot of red blood cells. It's not going to be rare for an NK cell, which is in the blood, to find a red blood cell, which is all over the place in the blood. Also, we know that we're alive with red blood cells. So our red blood cells must have all not gotten killed. So there's clearly something else going on here. And that is in fact true, because NK cells have this other type of receptor as well, which are known as NK activating receptors. And so not only do NK cells have these inhibitory receptors that are binding to MHC, they also have activating receptors that are involved in turning them on. This is our list of NK activating receptors. You can see that there are a whole bunch of different kinds. So you can see there's one CD16, there's some NKPs, there's some KIRs, there's some NKGs. We got a whole bunch of different NK activating receptors. This also shows ligands. And I want to show you the most important piece of this ligand part of the table, which is this. You can also see that there's a question mark there and there's a question mark there. And the moral of the story is that there's a lot we don't totally understand about these NK activating receptors. Sometimes we have some hypotheses about what's going on, but there's a lot we don't know. I can tell you broadly that NK cell inhibitory receptors usually bind MHC. I can't give you a usually for the activatings. There are still a lot of questions. And so instead of giving you a usually for the NK activatings, I'm going to give you examples of specific ones with the full knowledge that these are only examples and that they don't necessarily have anything in common other than that they're NK activating receptors. One of those that I want to tell you about is this one that's called CD16. And CD16 is also shown in the figure on the right of this slide. CD16 has this other name, which is that it's an Fc gamma receptor. What does an Fc gamma receptor bind to? Based on the name Fc gamma receptor. Yes, Jamie? An Fc region of an antibody. Specifically what? An IgG. So specifically, an Fc gamma binds to the Fc region of IgG. That means if we have some kind of cell with an antigen on its surface that has antibodies bound to that antigen, that can turn on our NK cell because there's this Fc gamma receptor that can be turned on by the antibodies. Great news. You already knew that. Because when we talked about antibodies, and I told you about the effector functions of antibodies, I told you one called ADCC, or NK cell activation. And it's exactly what I just told you. We have antigens on the surface of a cell. These antigens have to be on the surface of a cell because we're working with natural killers that kill things. If there was no cell, we couldn't do any killing. So we've got these antigens on the surface of a cell. Antibodies, specifically of the IgG class, will bind them. And then our NK cell, which has this Fc gamma receptor, CD16, will bind to the Fc portions of those antibodies. That will signal to the NK cell so that it will release its granules, its cytotoxic granules, and kill that cell. And so one kind of NK activating receptor is CD16, this Fc gamma receptor. So that's one way we could activate our NK cell. The other NK receptor that I want to tell you about in a little bit of detail is one called NKG2D. You can also see NKG2D here. And you can see a structure of NKG2D here. NKG2D has some specific proteins that it binds to. Those proteins are listed here as well as being on this slide. One of the proteins that NKG2D can bind to is called MIC-A. Another one is called MIC-B. Another one is called RAET1. These are all proteins that can bind to NKG2D. And that might seem like random. You're like, okay, fine. NKG2D binds to some random proteins. Whatever. But the key thing about those proteins is that they are proteins that a cell makes when that cell is under stress. So if that cell, for example, is infected by a virus and the virus is trying to mess with the cell's metabolism, the cell might make some cell stress proteins to indicate, hey, I'm under stress. If the cell is damaged in some way, it might make cell stress proteins. If we are talking about a tumor cell that is replicating super fast and super out of control, it might make some cell stress proteins. And so all of these cell stress proteins can bind to NKG2D, and this will turn on an NK cell in order to activate. And you can see this happening here. So if a cell, for example, is infected, you're seeing an example of a cell being infected, that might be stressful for the cell because of all the ways that the microbe is trying to mess with the cell. And so the cell might start making one of these ligands where it didn't before. Similarly, if our cell gets transformed and is a tumor cell, it might start making one of these ligands where the non-tumor cells weren't making them. So these are proteins that the cells only make under stress. And this will allow these cells to turn on NK cells. And so we can see an NK response here. And this is sort of the perfect example, though life is not always this ideal. So you might see a situation where we have a healthy cell. The healthy cell makes MHC class 1. The MHC class 1 inhibits the NK cell. We engage an NK inhibitory receptor and we inhibit that NK cell. Alternatively, we could see an infected cell that is missing MHC class 1 and that has cell stress proteins on its surface. And now we get signals through the NK activating receptor, and we activate the NK cell to kill this cell. This is sort of, like I said, the perfect situation. In reality, it's sometimes messier than this, because this is showing we have the inhibitor and no activator, or we have the activator and no inhibitor. In real life, sometimes cells will have some of both. And really what the NK cell is doing is it's actually kind of adding up its inhibitory and activating signals. If it mostly gets inhibitory signals, so you can see here it has 2 inhibits and 1 activate, the sum is inhibit, and the NK cell won't kill. The NK cell mostly has activates and no inhibits. So here we've got 1 activate, 0 inhibits, mostly on the activation side. The NK cell is going to kill. And then the sort of more complicated one is what happens when we kind of have multiple of each, and we add that up. And so here we're seeing we've got 3 activates and 2 inhibits. 3 is more than 2. And so we end up activating the cell. And so in reality, NK cells seem to be additive and adding up all of the signals they get to sort of help decide whether they're going to be inhibited or activated. Yep, Jamie.
Ever a point where it was
Given some of the things that we don't yet understand about NK cells, I don't know if we totally could answer that question.
okay?
So one thing that this implies is that NK cells actually don't have one receptor. Like a T cell receptor has 100 copies of the same receptor, that same one T cell receptor. A B cell might have 100 copies of the same B cell receptor. NK cells are different in that they have a whole bunch of different NK receptors on their surface. So we don't have just one kind of receptor on the surface. We have a whole big mixture. And even in an individual person, we might see situations like what you see here, where you have a bunch of NK cells and they all have different combos. Again, differences are sort of unclear here. How this works is a little bit unclear. If we look at the NK cell receptors in the genome, MHC class 1 molecules are the most diverse genes in the genome. So remember, you all have different MHC class 1 molecules. Turns out the second most diverse genes in the genome are the NK receptors. So you actually pretty much all have some different NK receptors too. It's not quite as diverse as the MHCs, which is kind of makes sense since MHCs and NK receptors can bind each other. It does seem like there might be some kind of like process that helps the cell know what's self and what's not. Again, we don't really understand what's going on. We don't understand how the cells are making choices of which ones to express. All of that is unclear. One thing that we do know is that I told you before that there are cases where different MHC molecules can be associated with good or bad outcomes following infection with different microbes. There are some situations where it's not just what MHC type you have, but actually what combos you have of MHC types and NK receptors. So, for example, if you have, say, HLA-C, and you have the NK receptor 3DS1, then you're going to have worse cervical cancer caused by HPV. Whereas if you don't have 3DS1 and you have HLA-C1, you're going to have decreased cervical cancer. And so you can see kind of all of this here. And most of these are something that is infectious, though you can also see there are some autoimmune conditions and a condition, preeclampsia, which is related to pregnancy complications, which you maybe would not think of as being a thing that would be related to immune-related genes. And so you can see that this does seem to have a lot of really important details. This also brings up a piece of information that is pretty important just in general. This also comes up more a little bit later in some other parts of thinking about immunology as well. But one subfield of immunology that is quite fascinating is pregnancy immunology. Because if you think about it, when a woman is pregnant, that fetus is half foreign. And if that same woman, before she got pregnant, if you just said, hey, I'm going to give you a kidney from your partner or whoever, that kidney would get rejected for being foreign. Yet somehow the fetus does not get rejected, even though it's also half foreign. And so in fact, pregnant women's bodies have to do a lot of modifications with their immune system in order to not reject a fetus. And of course, if this didn't work immunologically, we would not work. Right? So this is pretty key. In addition, some of your fetus's cells are missing your self-MHC. So there's also a missing self problem on some of those fetal cells. So we can talk a little bit more about pregnancy immunology as it comes up in some of the later places in the semester, particularly when we think about some of the things like transplantation. But one thing just to point out here is that one thing that we know is that the cells in the placenta, which are shown here in purple, are covered with things that hit inhibitory receptors. So basically, if any NK cell comes anywhere near the placenta, that placenta hits as many inhibitory receptors as possible so that that NK cell gets lots of inhibition. And so that NK cell does not actually kill the placenta. So you can see there's all sorts of different types of inhibitory molecules. One of them is another one of those things that sort of is like class 1, but is not class 1. It's called HLA-G. And it's just a protein that kind of looks like class 1, but inhibits NK cells. And the placenta is actually covered with HLA-G, along with many of these other things, so that it can turn off any of mom's NK cells that come near and try to kill that fetus. And so all of this is sort of pretty important applications of this information. So this is the info about NK cells as sort of, again, as I left graduate school, or as I was finishing graduate school, was what I've taught you so far. And I want you to tell me what you think about NK cells. Are they innate cells or are they adaptive cells? Are they part of the innate immune system or the adaptive immune system? If you were writing the textbook in 2008, would you have put them in the innate chapter or the adaptive chapter? And why? Yeah, Justin. So Justin just wants to split the difference and give it its own chapter, which kind of sometimes happens. But what if I said no, you can't do that. You only get 2 chapters. You only have your textbook only has 2 chapters in it. It's a very strange textbook. So you have to choose. Rishi. Okay, you're gonna put it in innate. Why are you gonna put it in innate? Okay, so Rishi's gonna put them in the innate chapter because they have an immediate response. All right, Sebastian. Okay. Okay, so maybe because they have some proteins that have similarities to adaptive, you're going to put them in the adaptive chapter. Jamie?
I would say adaptive, like solely from a learning point of view, you have to learn about things in the adaptive immune system, like MHC and all of that, before you can understand NK cells.
So Jamie would put them in the adaptive mostly because of the ways that you talk about them in relation to adaptive cells. Okay, I'll come back to that, yeah. Mm-hmm. Right, okay. So Sydney would put them in the innate immune system box. And it's a struggle, and it was a struggle, and it always was a little weird. By and large, people generally have put them, and still do put them, in the innate chapter. So usually we put them with innate immune responses. In fact, most people who are not me teach NK cells earlier in the semester with the innate stuff. But I feel like Jamie, that for learning purposes, I like to tell you about the MHC stuff, and I like to tell you about CTLs, and I like to actually relate it to things. So that's why I put them in a weird place. Even though if you actually go back and look at your textbook, they're in the innate chapter in your textbook. And so they're in chapters that they're in pages of chapter 3 that I had you skip or something that you're now going back to. So it's like a little weird, right? It's a little awkward. And this is one really obvious place where that nice, clear distinction of innate versus adaptive gets a little messy. And usually the reasons why people say innate, these are innate cells, is because they make the same response every time. They don't do VDJ, and they're responding early. That would be the reasoning that people would give for putting them in the innate chapter. And then things started happening. The moral of the things that start happening is that it got even messier. It's not that we had got an answer, it's that we actually got it even messier after this. Which is that there was a phenomenon described in 2008, 2009, of NK cell memory. And so the experiments that first showed NK cell memory are described in this figure as well as following slides of other figures from your textbook. So what was seen was that you could see some NK cells that responded to this infection with this virus called MCMV, or mouse cytomegalovirus. And you could actually see after those cells were responded to the infection, they went through a clonal expansion process, just like we see in the kinetics of our adaptive response. And then they went through a contraction response. And if you took those same cells and then tried to make them interact with infected cells in a dish, their responses were better. Their response had improved over what they'd had before. So they'd had that differentiation that we associate with an adaptive immune response. And if you actually transferred those cells into mice, and particularly you have to use some newborn mice that are really immunodeficient, those cells could protect the mice. You can also see this in this figure from your textbook, which specifically shows the clonal expansion aspect here. So here there were NK cells, and they were NK cells that had a protein on their cell surface that allowed us to track them in this mouse by flow cytometry. So we didn't have to worry about the mouse's own cells. We could just track these NK cells over time and their progeny. And then we infected this mouse with the virus. And you can see at the beginning, about 1%, there were about 1% of these cells. Day and a half after infecting, 1.88. 7 days after, 19%. 15, 3.9, or 13.9, 3.92, 1. It makes that perfect sort of curve that you think of for a primary response in terms of those cells undergoing clonal expansion and then contracting. And so that looks like an adaptive response. That's not what we usually think of with some aspects of the innate response. And then if they also did this experiment where they took those cells and put them into the immunodeficient mice with viral infection, we can see a few different things happen. So the blue line is what happens if you just give these baby mice virus and you don't give them any cells. And what you will notice is that they all pretty much die by 10 days of that virus infection. If you give those mice some naive NK cells that have never experienced infection before, and you give 10 to the fourth, so 10,000 of those NK cells. That's the red line. The mice do a little bit better. They all don't die. So about 25% survive, and you're seeing kind of them living for a bit longer. And if you give more NK cells, more naive NK cells, so now you're giving 10 to the fifth or 100,000 NK cells in the yellow line, the mice do even better. You give them more, better. Sure, makes sense. But then in the green, those mice are getting 10 to the fourth memory NK cells. So these are NK cells that have experienced infection before. And we're only giving 10 to the fourth. Remember, the red line here is what happened with 10 to the fourth naive NK cells. Now we do 10 to the fourth memory NK cells. Look at our survival. Look at how well those cells are able to protect. And so what we can see is that these cells are acting differently the first time they see the infection, in the red line, and the second time they see the infection, in the green line. These cells have actually improved their response or had this quality of a memory response. And so it seems as though we've seen this improvement of the response. And this is super weird because NK cells are not really thought of as being antigen specific. At no point did I really tell you with those receptors about antigens that we were specifically binding. And so it looks as though our NK cells can get some signals from receptors. They actually make cytokines like interferon gamma. They make perforin and granzyme. They proliferate, they contract, they make memory cells, they make a better response. They do all the things that we think of as part of an adaptive response. And so when these data came out, the whole question of are they innate or are they adaptive cells got a whole lot messier. And we started to realize that this was our sort of idea of innate responses in green, They look the same the first and second time with infection. This is what we used to think NK cell responses look like in the pink. And then you can kind of see a primary and a secondary response here. And now what we realize is that the NK cells also are making sort of a primary and secondary response. And you can also see sort of the myeloid responses look pretty similar. This has opened up sort of this new arm of immunology. And most of the time people talk about it as this arm of trained immunity, where in fact you can see many different innate cell types that change their responses after they've seen antigen. The specific mechanisms of how this works are a little different than what we see with adaptive immunity. This is really about epigenetics. This is about chromatin structure. Is the chromatin open or closed? And so this is just a very brief thing. So it's really about, are we changing how open or closed the chromatin is? And this can be seen in macrophages now, and in NK cells, and in quite a number of cells. And so we're starting to realize this, and we now refer to many different cell types as ILCs, or innate lymphoid cells. And we've learned that there are a whole bunch of cells that kind of sit on this messy border between innate and adaptive. And so as Justin said, now we've realized they're their own thing. We can actually subdivide ILCs into a lot of different types. I'm trying to find what I want, not really finding it. But you can see that we're starting to realize there are a bunch of different ILCs. They have some overlap with things like Th1, Th2. They are related to a number of things. So they are cells that have certain innate properties, but they come from the lymphoid lineage. So they are innate lymphoid cells, or innate lymphoid-like cells. There also is sort of this trained immunity that's coming from even macrophages and other NK cells. And so we've realized, again, that this border between innate and adaptive immunity is a little muddy. And we realize that, in fact, innate and adaptive immune responses have been co-evolving and sort of interacting with each other throughout evolutionary time. And so because of all of this sort of interaction back and forth, there are lots of gray areas between these two things. And they're not nearly as independent and perfectly different as we used to think, nor did innate stop evolving when adaptive came on the scene. We really have had the two co-evolving with one another. I'm going to start talking about peripheral tolerance and autoimmunity on Friday, and I will see you guys then. I will also see you in lab tomorrow.